So I have to say first thanks to ACS and to c&en and to the committee for selecting me for this recognition. And I'm very fortunate of being here and being part of this group. So the title of my talk is Chemistry to Enable Drug Discovery in Neuroscience and also Rare Diseases. But before I show you a little bit about my research, I want you to introduce you with my background. As many of the good things in my life, science to me came as a game. So when I was very little, I was playing with Lego sets, with getting my hands on chemistry sets, and I was also designing my first experiments in the backyard, building like trebuchets and catapults that my fathers allowed me to, or that my parents allowed me to do. And it was all of these experiences at the very start of my career that allowed me to get a sense of what the scientific method was. How do you design hypotheses, how you design experiments, and then corroborate this hypothesis. And also, sometimes when things don't work, you go and try to tweak your hypothesis and see and try to learn new things. And it was the thrill to discovery and learning new things, which inspired to follow science, and that instinct started growing and growing until eventually I decided to start a career pursuing something that allowed me to use chemistry to improve human health. And that's why back in Costa Rica, I started my PharmD. And during my pharmacy school, I was able to perform research in natural products. I was isolating new natural products and also characterizing their biological activity. And once I finished my PharmD, I knew that I wanted a more hands-on training in organic chemistry. And that's why I went for my master's. And here I was working with Dr. Perez and Dr. Lamoureux in doing the synthesis of these funny-looking molecules that you see here that they are called ferrocene naphthoquinones. And there I was interested in their antimalarial activity and also their electrochemical properties. And it was during these years that I learned and I get acquaintance of the field of medicinal chemistry. And I was very interested in learning more about that and get trained in this area, and that is why I went to Vanderbilt University. I flew to Nashville and I did my PhD in chemical biology. And then, to expand my chemistry skill set, I went to MIT for a postdoctoral position. While at Vanderbilt, I was working with Dr. Craig Lindsley, and I was lucky enough that in his lab, we are able to work in different subfields of organic synthesis. So I was working in total synthesis, in medicinal chemistry, and in the development of new methods. But I was particularly interested in medicinal chemistry. And here I was able to work in different projects and get my hands on and get a very good training. For my postdoc, I came to Boston. I was working with Dr. Timothy Jamison, and here what I wanted to do was something that was very, very different to what I did in my grad school. So I was exposed to synthetic methods that were something that I have never tried before, like photoredox chemistry and also flow chemistry. Flow chemistry is particularly challenging, coming from a total synthesis background and medchem, because in this one, you need to switch your brain. You need to switch your brain a little bit, because what works in batch might not work in flow, and vice versa. So this was a great experience to expand my chemistry horizons. But as I mentioned, as I give you a hint before, my passion lies at working at the interface of chemistry and biology. And here I'm very keen to two specific fields, which are chemical biology and medicinal chemistry. In both of them, they are similar in the way that they approach chemistry, in the terms that you use your chemistry expertise to manipulate biological systems. Specifically, I was interested more in medicinal chemistry because you manipulate the systems to develop new therapeutic agents, and that's in line with what I wanted to do in life, to create new medicines and help people. So when I started at Vanderbilt, I started to work in schizophrenia research. And schizophrenia is a very important public health issue, especially for mental health. And although there's several reasons, the most important for me is that around 50% of the patients, they don't receive the appropriate treatment. And most of the time it's because this treatment doesn't exist for them. And that is why at the Vanderbilt Center for Neuroscience Drug Discovery, we were working in different strategies to tackle schizophrenia from a different angle. And one of that different strategies is the mGlu1 receptor. mGlu1 stands for metabotropic glutamate receptor subtype 1. It's involved in many functions of the brain, but we were particularly interested in this target because in the literature we found that once you knock out the mGlu1 or the gene that encodes for mGlu1 in mice, they start suffering from a disruption in prepulse inhibition. And this is a feature that is characteristic in a subset of schizophrenic patients and also bipolar patients. Another piece of data that was really interesting in the literature was that this gene has a significant amount of SNPs, and these SNPs are enriched in schizophrenic and bipolar patients. And then in our labs, we established that these SNPs, what they cause for the receptor is that they decrease its functionality. So now that we knew that, we hypothesized that maybe if we create a therapy that targets the receptor function recovery, this will be therapeutically beneficial for this subset of patients. But at that time, we didn't have the chemical tools that allow us to activate the receptor and answer this question properly. And that is why I started a medicinal chemistry campaign. And starting with this heat, and after numerous cycles of design, synthesis, screening, and pharmacokinetic properties characterization, we developed optimized mGlu1 PAMs. And as during my introduction, it was said, so PAMs stand for positive allosteric modulators. And these ones are the activators that we needed to do the target validation studies. This is just a taste of the molecules of our first generation of mGlu1 PAMs. This compound 605. The compound is potent enough for in vitro studies, and also it's quite selective. It's quite functionally selective. And then when we test this compound in a series of different mutants, we found out that this kind of compounds can activate these mutants. So that's what we were looking in vitro. But we needed to establish if this effect in vitro translates to something that is significant in vivo. But when we tried to apply this probe in vivo studies, we find out its pharmacokinetic properties were not optimal because the compounds were unstable in plasma. And then I proceed to create a second generation of mGlu1 PAMs. And you can see here, going from, these are just some examples, going from green is good, yellow is bad. We get to compound 4909, that is a compound that is optimized in most of its properties. It has a good potency, and it has good physicochemical properties. And also very important for us, it has a much better in vivo pharmacokinetic profile. Now the compound is brain penetrant, and it also has a half-life in rodents of around 5 hours. And with this probe, now, we started to explore the biology of mGlu1 and its role in the brain and also in schizophrenia. We can say that mGlu1 activation decreased the dopamine striatal release, and that's a feature for antipsychotic activity. Also, in vivo studies, now we have demonstrated that mGlu1 activation attenuates amphetamine disruptions in prepulse inhibition. And this comes without affecting locomotor activity. So essentially, what we're seeing is an antipsychotic-like effect without sedation. And that's something that you would like in a treatment for schizophrenics. And this project, I'm very happy to say, that is moving forward at Vanderbilt. And that I learned a good deal of medicinal chemistry during these years. And also that treating schizophrenia as a genetic disease provide me a very good background for my current role at Vertex Pharmaceuticals. Here, I'm dealing with rare diseases. So a rare disease is a condition that affects a small subset of patients, but once you combine them all, it accounts for almost 10% of the population of the US, and half of them are kids, and that's really worrisome. There's thousands of rare diseases. Most of them have a genetic origin. They're hard to diagnose, and there are few treatments approved for these diseases. And that's why at Vertex, we believe in finding new treatments for these diseases. But we just don't want treatments that alleviate the symptoms. We want something that can treat the underlying cause of the disease, because that's as close as we can get to a cure. But in order to do this, we need to overcome some scientific challenges, because there's gaps in the biology knowledge of the disease. Sometimes we don't know exactly what the target is. And sometimes when we have a target, that these targets are hard to modulate. The mechanism of actions, they are not proven. And usually, we don't have a good chemical matter to start. But this is working in mostly like in an unknown area. But in Vertex, we believe that we can do it. And one example for this is the work of the company in cystic fibrosis. And in this disease, we have taken leverage about the knowledge of the biology of the disease, knowing the gene that caused the disease, the protein that is affected, and also how the mutants affect its function. And now the scientists have developed molecules that can improve the function of CFTR mutants. And the first example of that was ivacaftor, that is now a drug that is approved by the FDA, and now is also being explored in combination regimes to expand the treatment to a larger subset of cystic fibrosis patients. And just for my last slide, I would like to say that this is a really exciting time to do medicinal chemistry. There's always new discoveries in the biology side, and that comes with, that will enable new chemical approaches to treat diseases, new modes of action, new mechanisms of actions. And if you think about the tools that we have so far, they seem like more rudimentary. When you look at the future, we will have more advanced tools, more optimized tools, and this will, what will cause, I think, is that what we see on drug world today might in the near future not be, or might be druggable in the near future. And this will have a great impact to find new drugs that can change people's lives. And with that, I would just want to acknowledge my Vanderbilt colleagues, my MIT colleagues, and also my Vertex colleagues. All of them have played a really important role in shaping my career. Okay, thank you.